Hello children, in today's video we are discussing about the topic isomerism, reaction mechanism and the various reaction intermediates in the organic reactions. So first of all, our first topic is isomerism. Isomerism is the phenomena of existence of two or more compounds which are having the same molecular formula but the different properties. Such compounds are known as isomers and the phenomena is isomerism. So the isomerism is basically divided into two categories, the structural isomerism and the stereoisomerism. When we talk about structural isomerism, the compounds having same molecular formula but different structures. That is, the compounds having same molecular formula but different structures or manners in which the atoms are linked are classified as structural isomers. For example, when we talk about the various structural isomers we are talking about are chain isomers, then position isomers, functional isomers, metamers. There are variety of structural isomerism in organic chemistry. So first of the isomerism of the structural type is the chain isomerism. For example, let us take the example of C5H12 pentane. We can draw pentane directly in the linear manner as the normal chain, the straight chain. Now, we can make the chain a little bit shorter, the 4-carbon chain, and one of the carbon is in the form of the methyl, which is linked on the second carbon atom. That is 2-methyl butane. Now, the third case, the chain is still made shorter, and the 2 carbons are placed on the center part in the form of a branch, and it is known as 2,2-dimethyl propane. So all of these are the chain isomers in which the size of the chain is going on decreasing. The pentane, 2-methyl butane, and 2,2-dimethyl propane. Now coming to the second type of structural isomerism is the position isomerism. When two or more compounds differ in the position of the substituent atom or the functional group on the skeleton, on the carbon skeleton, these are known as the positional isomers and the phenomena as position isomerism. For example, when we talk about C3H8O molecular formula, the two alcohols that can be represented by it is propene 1-ol and propane 2-ol. The position of the OH functional group has changed from number 1 to number 2. So these are known as the positional isomers. In the position isomers, one more example that we can take is of but1ene. The position of the double bond has been shifted from the first carbon atom to the second carbon atom in case of but2ene. So both of these are considered as the position isomers and the phenomena as positional isomerism. Now coming to the third category, that is the functional isomerism. In case of functional group isomerism, two or more compounds having the same molecular formula but different functional groups are known as functional isomers, and the phenomena is termed as functional isomerism. For example, if we are having the molecular formula C3H6O, so it can be represented in the form of aldehyde as well as a ketone. For example, the 3-carbon aldehyde, it is propanol. The formula is C3H6O, but it can also be represented in the form of a ketone, that is CH3COCH3, that is acetone or propanone. So both of these are having the same molecular formula, but the functional groups are different. One of them is having the aldehyde and the other is the ketone. Now the second one, the other example we can consider here is that is CH3COOH. That is ethanoic acid. And similar to it, the other functional isomers we can draw for it is HCOOCH3, which is acetate methyl methanoate. This is methyl methanoate. This is the ester. And this one is the carboxylic acid. So both of these are the functional isomers having the same molecular formula, but the different structural formula and the functional group. Now let us come to the other isomerism, that is the metamerism. In case of metamerism, it mainly arises due to the different alkyl chain on the either side of the functional group in the molecule. For example, C4H10O represent methoxypropane, that is CH3OC3H7, or it can also represent it in the form of diethyl ether. The various forms for it can be, that is, we can draw it like this: CH3O, CH2, CH2, CH3, or we can draw for it the other way as C2H5O, C2H5, or again we can draw it in the other manner as CH3. One of the other form that can be drawn for it is CH3O. In the form of the isopropyl linkage, we can also show it. Now, these are all considered as the metamers for each other. So, in the case of the metamerism, there are the different alkyl groups. Now, here comes the stereoisomerism. When we talk about the stereoisomerism, then the compounds that are having the same constitution and sequence of covalent bonds, but there is difference in the relative position of the atoms or groups in the space, that is, we are talking about the 3D space. These are considered as the stereoisomers, and the phenomena is known as stereoisomerism. The stereoisomerism is further classified into geometrical, optical. The various forms of isomerism can be classified for it. But for the stereoisomerism, we will be doing in the next part. But in this one, we are talking about the structural isomerism. So a brief outline of the flow chart about the isomerism is, the isomerism is divided into two parts, the structural and stereo. Structural again chain, positional, functional, and metamerism. There is also one other isomerism. This is known as the tautomerism. This is a special kind of functional isomerism in which the isomer exists in dynamic equilibrium with each other. The isomers are having a dynamic equilibrium with each other. And it can be further considered as a dyad system and a triad. In the dyad system, it involves the 1-2 migration of the proton. 1-2 migration, that is, the H has been shifted from this place, HCN or HNC. In the second is RCN, RNC. And the other form of tautomerism is triad system, in which the movement of proton is taking place in the 1-3 system. That is 1, that is the CH3 to O. Movement is taking place like this. For example, the one is ethanol, CH3COH. The H is moved from CH3 to O part. So it becomes CH2 double bond COH and H. This is called the vinyl alcohol. And in the other case, we have taken the example of propanone, CH3COCH3. And again, there is a 1-3 migration. The proton has moved from CH3 to O. So it becomes CH2 double bond C, OH and CH3. That is prop 1-ene to all. So these are the other one, which is in a brief description, it is known as a tautomerism. The four main types of the structural isomerism are chain, position, functional, and metamerism. Now let us come to the other topic of this video, that is the other part is based on the how the reaction proceeds, that is the mechanism part. In the fundamental concept of organic reactions, it is considered that when the organic molecules undergoes treatment with the attacking reagent, then an intermediate is formed. In between the reaction, the intermediate is formed, which further results in formation of products and the byproducts. So what are these reaction intermediates? These reaction intermediates are the short-lived. This is very important. The reaction intermediates are very highly reactive, short-lived chemical species through which majority of chemical reactions takes place. Many of the chemical reactions get accomplished only when they pass through the reaction intermediates, and these can be the carbocation, the carb anion, the free radicals, carbenes, nitrines. There are various types of reaction intermediates which are formed between the reaction that further results in formation of products and byproducts. Now the third main thing is the reaction mechanism. In the reaction mechanism, what we came to study is, this is a sequential steps. These are sequence of steps which helps us in describing how the electrons are moving, what are the energy changes that are taking place, how the bond is cleaved, how the new bonds are formed, the rate of reaction. When we came to know about each and every detail of the reaction, it is known as reaction mechanism. And for the reaction mechanism, we must be knowing how the bond has undergone the cleavage or a fission. So a covalent bond can be cleaved in two ways, the heterolytic cleavage or the homolytic cleavage. In case of heterolytic cleavage, the bond breaks in such a fashion that the shared pair, that is the covalent bond, moves to one of the fragment. And this fragment which we are talking about must be highly reactive in nature. It must be highly electronegative in nature. So it will take away the bond on it, and the other one will become electron deficient in nature. So the one from which the bond has been taken is now existing as sextet of electronic structure. That is, it is having the 6 electron, while the other one is having the number of electron pair more in nature. It is having one lone pair, as the whole of the bond has been taken away by the other species which is electronegative in nature. So in case of heterolytic cleavage, the one main thing that is found is one is having the less electronegative nature and the other which is more electronegative takes away the bond on it. Due to which, if these are the carbon containing, then if in case of the heterolytic cleavage, the carbon gets the positive charge, it will be known as carbocation, while the one which is having the less number and it is a carbon species, then it is called as carbocation. But if the carbon is getting the lone pair or the covalent bond on it, the shared pair on it, then it will be known as the carbanion. So let us discuss these two reaction intermediates in between. In the heterolytic cleavage, the bond breaks and the electron comes on one of this and the sextet of the electron is maintained. So this bond is shifted on the one which is more electronegative, that is Cl. Cl becomes negative and this is third butyl chloride, tertiary butyl chloride. And since chloride ion, that is the Cl negative, is more electronegative, so the covalent bond is shifted on it. This is third butyl carbocation. Butyl carbocation. The carbon is getting the positive charge. This is the carbocations are further classified as 1 degree, 2 degree, 3 degree based on the number of the alkyl parts attached to it. If the carbon which is having a positive charge is having alkyl group on one side, it is known as 1 degree carbocation. That is the ethyl carbocation. It is the ethyl carbocation. Now the other one which is the 2 degree and drawn, this is the isopropyl carbocation, and the last one, that is the third carbon which is having the 3 carbon methyl group on 3 sides. This is known as the third butyl carbocation. So the third one, that is the third butyl carbocation, is highly stable in nature. And why it is stable? It is due to the inductive effect and the hyperconjugation. Both of these will be discussed a little bit after the whole of the reaction intermediates must be explained in beginning. So third butyl carbocation is going to be most stable 3 degree, then 2 degree and the 1 degree. So these are the most stable carbocation, and the two reasons behind it is one is the inductive effect and the other is hyperconjugation. So these are the form in which we can generally represent a carbocation. When there are 3 R's, it is 3 degree carbocation. If there are 2 alkyls, it is 2 degree carbocation. If it is 1 R, then it is 1 degree carbocation. And if it is simply CH3 positive, then it is known as the methyl carbocation. Now, these carbocations are highly unstable, reactive. They are having a positive charge. They are sp2 hybridized in nature. One of the p orbital is vacant, that is empty. And on the 3 side, there is the alkyl group present. So this is the main carbocation, the intermediate. But the other thing that we have studied is if the carbon gets the covalent bond on it after the cleavage. So in that case, it is known as carb anion. These are also unstable intermediates, and in this case, these are only obtained when the bond reactions takes place in the ionic or a heteropolar nature. So let us assume that a hydroxyl groups attacks on a ethanol part. So the OH negative takes away this H and becomes water, and the bond is shifted on CH2, that is the carbon of the aldehyde, the aldehyde CH3CHO. So the carbon gets the negative charge on it, the bond shift on it. So the carbon becomes negative and it will be known as the carb anion. So in case of carb anion, one thing to be remembered is that the methyl carb anion is going to be highly stable in nature. So this works totally opposite as we have done in case of the carbocation. In case of carbocation, the 3 degree are the most stable. But in case of carb anion, the methyl carb anion is going to be the most stable in nature. And the main reason behind it is when there is increase in density of negative charge on the carbon. Due to the 3-methyl, the methyl groups are, remember here that the alkyl groups are electron releasing in nature. They are electron releasing groups. So they increases the electron density. So if the electron density on the carbon increases very large, it results in destabilizing the carb anion and thus decreases the stability. So the methyl carb anion is going to be highly stable in nature out of all of these. Now in case of homolytic cleavage, in the case of homolytic cleavage, one of the electron shared pair is equally divided among the bonded atom. So there is a movement of single electron on both of the atoms which are attached, and it is shown with the help of a fish hook. So look here, where we have taken the example of ethane, the single bond in between CH3 and CH3 has equally distributed. There is a homolytic cleavage, homolytic fission takes place, and the bond is equally divided in both of these parts. So each of the methyl is getting a dot or an electron, and these are known as the methyl free radical. Okay. Now alkyl radicals, these alkyl or the free radicals are further classified as the primary, secondary or tertiary in nature, and their stability follows the same pattern as that of the carbocation. For example, in the case of the 3 degree free radical, it is going to be most stable, then 2 degree, and then the 1 degree. You can see the structure of the free radical also, the carbon. On 3 side, there are the alkyl group. Again, it is sp2 hybridized, and the bond angle is 120 degree. But the main difference is there is presence of one unpaired electron in the p orbital. So we have discussed about the carbocation, the carb anion, and the free radical, which are obtained during the heterolytic and the homolytic fission of the bond. Now, other than these, the two important terms that we go through during the organic reactions are electrophiles and nucleophiles. So let us move with this topic, electrophiles and nucleophiles. Electrophiles, these are the species which are electron loving in nature, and as they have the presence of electron deficiency atom in it. Why they are electron loving in nature? They need electron because they are having electron deficient atom in them. For example, H positive, Cl positive, alkyl positive, BF3, which is a 6 electron species, AlCl3, they all are considered electrophiles. And the nucleophiles are those which are nucleus loving species or are electron rich species. For example, H negative, Cl negative, Br negative. So either they are usually having the negative charge, they are electron rich, or they may be neutral in nature, having a lone pair on them. But the main things that we have to remember while we are doing the electrophiles and nucleophiles is that all the positive charge species cannot be considered as the electrophile. But only those positive charge species which can accept a pair of electron can act as a electrophile. For understanding this term, let us take the example of H3O positive, Na positive, Ca2 positive. Although they are having the positive charge, but they are not electrophilic in nature. They are not considered as electrophiles. Now the other important thing, Nucleophiles always attack the substrate molecule at the site of lowest electron density because nucleophiles are electron rich. So where they will attack? They will attack on the site where there is less electron density. Now the last very important point, electrophiles behave as Lewis acid. So Lewis acid we will study about this term in equilibrium. So Lewis acids are those which are having a strong tendency to attract the electrons. And what are the Lewis bases? Which are having the strong tendency to donate the pair of electron. The similarities with the nucleophiles. So nucleophiles are considered as the Lewis bases and electrophiles are considered as the Lewis acid. So dear students, in this video, we have gone through the various types of isomerism, structural isomerism, the various reaction intermediates, then the electrophiles and nucleophiles. So now in the other one, we will be meeting with the various types of movements that are taking place during the organic reactions. For example, the resonance, the electromeric shift. So till then, revise the video, stay safe, stay healthy. Goodbye.